Alright, welcome to chapter 24-1. Here we'll discuss the general anatomy, the general function, and the histology of the digestive system, and then we'll talk about how we actually control and move parts of the digestive system. So let's go ahead and get started. On the left here, I have a picture from The Awkward Yeti showing one of my favorite depictions of the digestive system, starting with a piece of pizza at the top and eating it, and then using some teeth to churn it and break it down, and then the tongue shoveling it back into the esophagus. The esophagus starts contracting and moving all of the food into the stomach. Stomach provides some acid, then dumps it into the small and the large intestine. And at the beginning of the small intestine, we have the liver on the right, dumping in bile that is stored by the gallbladder, which is to the right of the liver. And then to the left, we have the pancreas in yellow, and it's donating some enzymes into the small intestine to help break down some foodstuffs. And then the small intestine turns into a large intestine, and then that finally goes into the toilet. I really like this picture because it's cute. You can see the little appendix on the bottom left corner. The only thing I would change about this picture is the pancreas should really be on the right side of the picture, left side of the patient, and flip that with the liver and the gallbladder. That's my only negative to this picture, but I think it's a really good, cute, short version of what we'll really talk about. So as per usual, we have our Can You Describe questions for 2 slides to use for studying. And starting off with the anatomy. So the digestive system is also called the alimentary system. You might also hear it called the gastrointestinal tract or GI tract. You might hear of GI docs. These are docs that are specializing in the GI tract. We can think of the digestive tract as a giant muscular tube. So from beginning to end, a big, long tube made of almost entirely smooth muscle. And this is actually part of how we developed. So when we were first a tiny ball of cells as a zygote, then we started differentiating into different types of cells. So we started differentiating those cells. So that ball of cells then moved some of those cells into one quadrant and then left a little bit of space. And then that eventually turned into maybe making 2 spaces and moving all of the cells into other portions. And this all depends on what species you are, and frogs differentiate a little differently than dogs and humans and so on. But essentially we keep making these holes. And one of the first holes that's made is the beginning of the GI tract. So you can either start with the mouth first, or you can start with the anus first, and essentially build the cells around this tract during development. And if you start mouth first, there's a term for that, but we actually start as humans anus first, and this is called a deuterosome. Deuterosome. I probably have that misspelled. But this is fun facts for 500. So anyone who says they're talking out of their butt, that probably originated from this concept. Anyway, in the picture you can tell, or on the left-hand side, they list all of the major organs of the digestive tract, which we'll go through independently in the next section. And then to the right, we have the accessory organs of the digestive tract. And these are going to be really important in helping actually digest the food. The major organs of the digestive tract are really more breaking down and moving things along, mechanically breaking down and moving things along, while the right side is more like chemically breaking things down and moving things along. So functions in general, we're breaking down food, we're absorbing nutrients, and we're eventually eliminating them as waste. If we were to separate out the 6 individual functions in order of how they occur, we can start with ingestion. So this is food in the mouth. We're actually thinking about what food we're putting in our mouth. We find the food and then we put it in our mouth. Then we have mechanical digestion. This is where we're chewing and we're crushing and we're tearing apart larger pieces of food into smaller pieces of food. And then propulsion is actually moving it down the tract. So either from the esophagus to the stomach or from the stomach to the small intestine. And then we have the chemical digestion. Some of this happens in the mouth, but the majority happens in the stomach and in the small intestine. And we'll talk a lot about that. Secretion, so what are those accessory organs secreting into the GI tract so that we can further break down those foodstuffs. Then we have absorption, and absorption is the whole point of eating. So how can we get those nutrients in their smallest form so that we can most easily absorb it by the tissues and then eventually into the bloodstream so that it can circulate and go to the appropriate cells so that they can use them however they need to rebuild other molecules in the body. And then finally, last but not least, defecation or poop. Feces. Super fun. All right. So additional functions of the digestive lining. So just the lining as it's moving the foodstuffs along. It's protecting against corrosive materials. So we're thinking not only the corrosive materials you might eat that might be really acidic or really basic, but your stomach is going to provide a lot of acidic components, and that acid can really deteriorate the epithelial lining. So most of the GI tract is actually lined in a mucous membrane to help protect the epithelial lining superficial to that. We also want to protect against the mechanical stresses. So, your teeth are chewing up the food and all of that food goes down the esophagus. But if the food is a little too large for the esophagus, how can we still maintain the integrity of the esophagus as we're moving food down? How can we maintain the integrity of the sphincter muscles in order to help segregate the food from one organ to another? How can we help the stomach contract without ruining any of the organs that are surrounding the stomach, etc., etc.? We also need to protect against the bacteria that are swallowed with food or that permanently already live in the GI tract. So bacteria that we may swallow, they might get destroyed either in the mouth, but more likely in the stomach. But most importantly, we need to talk about something called the microbiome. And the microbiome is, you might have heard this in the news or in popular health articles, but this is a big area of research right now because the microbiome is a heavily studied topic that we actually don't know a lot about. We generally understand how it interacts with humans, but we don't understand how we can kind of manipulate these interactions in order to help better people. So what is the microbiome? Essentially, there are a bunch of, and here comes a terrible picture of a person, of a human, who's happy to not have a neck, I suppose. Anyway, so the microbiome. So we have a bunch of bacteria living on us at any given period of time. And the bacteria that live, like let's say near your belly button, are going to be very different than the bacteria that live near your pelvis, that are on your feet, that are on your hands, on the front of your hands versus the back of your hands, on your face, on a good skin day, on a bad skin day, etc., etc. There's also bacteria that lives inside of your gut, so that's actually living within the GI tract, so that entire tube. And we consider this good bacteria, and usually for the most part it is good bacteria, and we call this a that's supposed to be 2 M's, commensu coronavirus. Great. I don't need any more news about that. Commensalism. So the good bacteria lives in harmony with your cells. So for every one human cell that you might have, so think one single epithelial cell, one cardiac muscle cell, one bone cell, you'll have 10 bacterial cells. And this is a crazy number to think about, but it really is true. For every one human cell, you'll have 10 bacterial cells that are living on or within you. And we have a commensalistic relationship with them, meaning that we live in harmony with them. So your body is recognizing self versus not self, but is determining that not self is not a threat, and so they don't activate the immune system to attack it. When it does activate the immune system to attack it, and maybe the bacteria is good bacteria, then we have problems. If the bacteria is not good bacteria, then we want the immune system to attack it. But this relationship is happening all of the time. And this bacteria is also really important in the gut because it actually will help assist in breaking down the food and helping you utilize the best part of the nutrients that you would need. So this relationship has been existing for years. Research is really interested in moving microbiomes from one person into another person. There's some good studies that have looked at people with diabetes versus people that are not diabetic, and moving microbiomes from those that are not diabetic into those that are diabetic, and it actually has helped with insulin production and or weight loss. There are interesting studies. People who are related will have more similar microbiomes than those who are not related. People who are not related that live together in the same house over time will develop this almost similar microbiomes. Women versus men living in the same place. Women will be more likely to have microbiomes similar to women than those of the male counterparts, even within the same household. So there's a lot of and even if you have a pet, that changes your microbiome. When you're pregnant, that changes your microbiome. There's a lot of really interesting research around this topic. I suspect within our lifetime we'll be able to see a lot of really good changes with inflammatory bowel diseases and things like Crohn's disease, and being able to better differentiate the different types of disorders that folks have and how we can better treat those. So anyway, stay tuned. All right, so let's talk a little bit about what's going on in the abdomen. So let's zoom in on the thorax, and here are some legs. Maybe here's our respiratory diaphragm. This is right, this is left. So let's draw in some organs. Here's going to be our liver. Here is going to be our stomach. Our spleen is going to be hanging out over here. We've got some kidneys in the background, a pair of them. The small intestine is connected to the esophagus superiorly, and then the intestines. So we have beginning the small intestine first, so SI, and then that turns into the large intestine, and then that finally exits out of the colon. So I'll put large intestine. We also need to add in some organs like the pancreas is over here. We've got a gallbladder over here, so GB. Why do I feel like I'm missing one? Oh, we have the appendix that's hanging out over here somewhere. I don't think I'm missing anything. It feels like I'm missing some. Oh, well, the adrenal glands are out over here. Okay. Anyway, so remember that this is what we're dealing with in the abdomen. And so we have to have a way to protect all of these organs and separate them from themselves. But it's part of the digestive tract, so they need to have a way to communicate with each other too. So we need to separate them, but also allow them to still have some amount of communication with each other. So developmentally, most of these organs, virtually everything but the kidneys, develop together, and the kidneys develop separately. It's a little bit of an oversimplification, but for the purpose of understanding what the mesenteries are doing, this is a good start. And because of that, everything that I've circled in orange is going to have its own set of lining, and then the kidneys are going to be behind that lining. So we're going to call the kidneys are going to be retroperitoneal. You probably remember this. I started talking about this whenever we opened the cats for the first time. You can appreciate that you saw this like shiny membrane, and you had to peel back that shiny membrane in order to actually expose the kidneys. And that shiny membrane is going to be the peritoneum. And that peritoneum, since the kidneys are behind it, we call that retroperitoneal. Okay, so what are these mesenteries? So we have the peritoneum that's circulating the entire abdominal cavity. And you can kind of think of the mesenteries of like a sack of potatoes. So if I have the sack, that's going to be my peritoneum. And then I'm going to have my organs within it. So I'm just going to draw circles. Okay. And just like what we've learned with the lungs and the heart, each one is going to have its own visceral lining. So we're going to call that visceral peritoneum. And then each one is going to have its parietal peritoneum. And the parietal peritoneum realistically isn't this close to the organs. It's actually a little bit closer to the to the peritoneum than it really is to the organ. And just as we've learned with other organs, we're going to have some fluid that'll separate out these layers. Okay, so I have my sack of potatoes, and then I have my potatoes, and my potatoes have potato skins, and they're surrounding in some sort of magical fluid that's in my sack of potatoes. But now we have to have a way to make sure that my liver potato sits up high and my large intestine potato sits down low. So how can we strap these in to the bag, the peritoneum, so that they don't float around and bump into other things? And that's where we actually need to add in the mesenteries. So I'm going to redraw this picture. And now we're going to draw in the mesenteries. And mesenteries are essentially two layers of peritoneum. So if you can envision the peritoneum folding in on itself and grabbing an organ with it, that's a mesentery. And if we have 4 layers of mesentery, let me do this in a different color. Then that is going to be called omentum. Okay, so again, if we just double the doubled layers, then we get omentum. The mesenteries are super important because within the mesenteries, we're going to be carrying important arteries, veins, and nerves to and from the organs. And it's nice because within these 2 layers of peritoneum, we're also going to have some adipose tissue to help insulate and protect, or insulate insulate these arteries and veins, and to also help protect those arteries and veins. The omentum is going to have even more adipose tissue, and some arteries and veins, not really a lot of nerves, but it's mainly purposed to be a lot of adipose tissue. So this is going to be more of a protective feature. This is more of a organizational feature. Okay, so let's draw these in. So in purple, I'm going to have my mesentery, and my mesenteries are going to strap in 2 important items. One is going to be the small intestine, and I'll shade that in. And then the other is going to be the large intestine. And again, we're going to have our arteries and veins in here. Okay, so you saw this whenever you were dissecting. You just didn't realize that's what it was. But all of that shiny material that was holding the small intestine together and keeping it one big ball of tissue, those were the mesenteries. And I asked you to kind of pull apart the pancreas outside of that mesentery, but still leave some of the mesentery behind, because those mesenteries are actually helping hold in the small and the large intestine. So that's why I didn't want you all to remove it. And all of those mesenteries, they actually strap in to the posterior wall of the abdomen via what we call the mesentery proper. So both the mesenteries from the small intestine and the mesenteries from the large intestine both meet at the mesentery proper to adhere to the posterior wall of the abdomen. So that's how we're strapping in the small and the large intestine. Now we need to talk about omentum, and this is how we're going to be strapping in the stomach. So let me draw the stomach down here. And the stomach is J-shaped, as we'll talk more about. So that's a terrible picture of a stomach. Let me write stomach. And the stomach has 2 curves to it. One is called the lesser curvature of the stomach. and the other one is called the greater curvature of the stomach. And the on the lesser portion or the lesser curvature of the stomach, we're going to have one layer of omentum, and this is going to be called the lesser omentum. And then on the opposite side, we'll have more omentum, and we'll call this the greater omentum. All right, you probably didn't notice the lesser omentum very much during dissection, but you definitely noticed the greater omentum because this was that big, long, sac-like thing of fat. And you were like, do I cut it? Do I remove it? Why is this here? So this greater omentum is going to lay over on top of the entire abdomen. And so in this picture, I'm going to draw it like this. It lays over the entire abdomen in order to protect those organs anteriorly. And then the lesser omentum is helping actually hold in the stomach in place. Lesser omentum kind of helps the liver as well, but we don't talk about that in this class. Okay. Greater omentum. I told you it has a lot of adipose tissue. So of course that's going to be insulative or provide insulating properties and also protective properties. But let's talk about some not so helpful pieces. So we have what we call visceral fat. And we also have something called subcutaneous fat. So the visceral fat is actually the fat that exists in this omentum. So if you're packing on a little weight in this area, it might be visceral fat. Then there's also subcutaneous fat. So subcutaneous, sub meaning deep to the skin, cutaneous. And so this is fat that's actually sitting on top of the muscle. So if you have your rectus abdominis here, your subcutaneous fat, so your visceral fat would be behind it in yellow, and then on top of it you're going to have your subcutaneous fat. And so why is this important to differentiate the two? Well, men tend to carry their weight in visceral fat. This is where we get like a dad bod or a beer belly type look, versus women tend to carry most of their fat in subcutaneous fat, and this is why it's not evenly distributed. I'm sure there's some evolutionary reason why. I'm not necessarily well aware of it, but this is why it's very difficult to exercise and isolate the effects of your exercise. So if you're doing crunches all day, every day, you probably won't really be able to make much of a difference of the amount of fat here, because you're not really targeting this fat over this fat, or this fat over this fat. It's a systemic thing. You can't isolate which fat you're targeting, unfortunately. So anyway, okay. Back to the mesenteries. All of the, and then how do we strap in the liver? We haven't talked about the liver. So I'm going to move back up to this picture. How do we strap in the liver? So our liver's over here, and it's the only thing that really we haven't talked about. I know I haven't really mentioned the spleen, but the spleen directly attaches to the greater omentum, which is attached to the stomach, so that's fine. But the liver has its own ligament, and this is a remnant of a piece of tissue during development that we don't need to worry about. But we call this ligament the falciform ligament. Falciform ligament. And this is also what's helping strap into the posterior aspect of the abdominal wall. A little bit inferior to the respiratory diaphragm. Okay, so here's a picture where I have all of the terms listed on the left, and then on the right we have a sagittal view. Right is anterior, left is posterior. Showing in different colors all of the mesenteries and where they're attached. I just think that this is a really difficult view to really understand this topic. So whatever view makes sense to you. There's also another view in these next two pictures. Over on the left is showing what's going on developmentally, but on the right I have boxed out the items that you'll need to know and in relation to the large intestine and the stomach. Keep in mind that some things are hidden from this view. It's just isolating the organs involved that are attached for the omentums and the mesenteries. Same thing going on here. This is a little bit of a deeper view, showing the peritoneum. But again, I think this can be a little confusing if you're not already familiar with what the abdomen looks like. So hopefully one of these views works for you. If not, reach out to me and we can chat. Okay, we mentioned this. And all right, so now moving on to the histology, our favorite. So as I said, the GI tract is one giant tube from mouth to anus. And within this tract, if we were to zoom in and look top down, we would have 4 distinct layers. So one layer, and another layer, and another layer, and another layer. Throughout the entire GI tract. So the entire GI tract will have all 4 of these layers. In some areas, we might have a little thicker version of one layer versus another layer. In another area, we might have some specialized glands in one layer, but not in another area. So it's important to understand these 4 layers before you start learning what's going on at each individual level. So the 4 layers, from deepest to most superficial, you have the mucosa, or mucosal layer. Then we have the submucosa. Sub because it's deep to the mucosal layer. So even though this is a tube, we're thinking from the inside out. Then we have a muscular layer to help us move stuff. And then finally we have an outer layer called a serosa layer that just provides protection for the muscular layer and maybe some points of attachment. That's about it. We have some specialized areas of the GI tract, and we mentioned some of these now. So the stomach also is going to have additional longitudinal folds, which just allow for expansion of the stomach in case you have a larger than normal meal. And then the small intestine has circular folds, which allows for expansion during distension of the smaller and large intestine, during distension of the small intestine as you're moving things through. This is really important because we need to increase the most amount of, or we need to be able to increase surface area as much as possible in order to allow for absorption. So here's a nice cartoonistic picture. On the left, you've got your tube, and boxed in red, I have submucosa, and then to the right, a zoomed in version of the submucosa. I also have all the items that are identified just in the submucosa. Same thing with the muscular layer in blue, and then the names of the muscular layers in blue on the right, with the myenteric plexus in the middle. We'll talk about that here in a second. What the picture originally is depicting is in that box. So you have the tube, and then this box is zooming into one of the villi, and then those villi also have, the circular folds. And on the circular folds, we have microvilli, and so that also is increasing the amount of surface area that we get exposed to, or that we have the option to use. This image will be really important for you to study with, so please be familiar with this. All right, so let's talk about each layer independently. Starting with the mucosal layer. The mucosal layer has the simplest and thinnest types of cells. So we have simple and stratified squamous. This is in most of the areas of the GI tract. Then we've got some simple columnar epithelium. This is really restricted to the stomach. That'll make a lot more sense whenever we actually talk about the gastric pits in the stomach. And the shape of the gastric pits really only allows for the simple columnar epithelium to fit in there. And that'll be really important whenever we talk about how the mucosal cells and the enteroendocrine cells are working. And we also have some goblet cells sandwiched in between all of these cells in order to be continuously secreting mucus. So just like we saw in the respiratory system. Then in the lamina propria, just like with every lamina propria, we've got blood vessels, we've got nerves, we've got lymphatics, and we've got a little bit of smooth muscle. The smooth muscle here is not the primary smooth muscle we'll really talk about, but there is a little bit of control from these muscular layers in terms of moving things, but it's more just for shape rather than actually moving things down the tube. And Then we have the muscularis mucosa. This is all of that muscular layer, and we'll have not only circular layer or circular shaped muscles going down the tube, but we'll also have longitudinal shaped muscles going down the tube. Again, this is not where we really care about the muscular layer. There's just a little thin layer of muscle here that we want to address, but we'll talk about the same shaped muscles in the muscularis layer. All right, the next deepest is the submucosal layer. And here what's really important is we have our let me do this in a different color. We have our submucosal plexus, nerve plexus here. And this plexus is important for local control of what's happening in the small intestine. So as the environment becomes really acidic or maybe really basic, or we have a hormone floating around, or we have some angry bacteria and we need to call on the immune system, the submucosal plexus is going to be one of the first that responds, which makes sense because this is the second most close to the contents of the lumen of the tube. So they're going to be the first ones that even get sense that there's any bad things happening in the tube of the GI tract. In the submucosal plexus, just like with every I shouldn't say every nerve plexus, but with deep nerve plexuses, not like arm and leg, we've got sensory neurons, we've got parasympathetics, sympathetics, and we've got postganglionics. So postganglionics coming from the autonomic nervous system. All right, then we have the muscular layer. Let me draw this in red. The muscular layer is the one where we really care about what types of, or the orientation of the muscle layers. So again, we have circular and longitudinal muscle layers. So circular, meaning they go in this direction, all across the tube. And when these muscles contract, they're going to constrict the contents of the tube. So this will allow for constriction of the tube or relaxation of the tube. Versus we have the longitudinal muscles, and let me do that in pink, that goes down the tube. And that is going to actually help move things down the tube. So we can squeeze things down a really tiny hole, or we can squeeze things down a thicker hole or a wider hole. But how do we actually help propulse things down the tube? So you can imagine as these contract, right, the length of the tube gets shorter, then it kind of inches its way along the track, kind of like an inchworm. These muscles have to be controlled by something, and so they are controlled by what we call the myenteric plexus. The myenteric plexus is important because this is what's engaging in short reflexes. That depending on the amount of distension in the tube or the amount of stretch in the tube, they will respond by initiating more contraction so that we can help move more of the materials down the tube. Where is the myenteric plexus really stemming from? Well, this is branching off of something called the enteric nervous system, ENS, enteric nervous system. I don't believe you guys have talked about this during 201, during the nervous chapter, because this is something that's usually not really considered mandatory to understand in A&P courses, and it's also something that we also don't really know a lot about. But this is actually a branch of the of the peripheral nervous system. So we have central nervous system, which is brain and spinal cord, peripheral nervous system, which is everything else. And the peripheral nervous system is motor, sensory, and autonomics. Motor, sensory, and autonomic nervous system. And then the autonomic nervous system, we've got sympathetics and parasympathetics. But what they also don't teach you, or maybe they did talk about it, is the enteric nervous system. So parasympathetics, sympathetics, and enteric nervous system. So we think of parasympathetics as rest and digest, but the enteric nervous system is really what's coordinating most of what's going on in the abdomen. We just don't talk about it because it's, like I said, we don't know enough about it. In human cadavers, you'll notice the enteric nervous system is a huge strangly webbing of nerves, and it's a lot. It honestly really looks like something that Spider-Man like shot out of his arm onto a wall. It's crazy. We don't expect you to really know the details of the enteric nervous system, just that it exists and where it is and where does it belong as a part of this whole nervous system flowchart. So now we have parasympathetics and we have sympathetics in the GI tract. So just like what we've already talked about, sympathetics will promote contractility, parasympathetics will decrease contractility. This is important because parasympathetics need to be turned on so that we can have more absorption happening, versus sympathetics need to be turned on in order for us to move it along and get to the next piece of food stuff that we can actually absorb. All right, last but not least, then we have the serosa layer or the serosal layer, either's fine. This is just, again, a protective layer for the entire GI tract, and it has some collagen fibers so that we can strap into other parts of the peritoneum. Nothing too special with the serosal layer. Pictures. Here we have a tube, or the part of the GI tract, and the different layers are exposed, so you can appreciate the serosal layer as the most thin, translucent piece in this picture that joins in with the mesentery. Then you have the muscular layer, so you have the outer longitudinal layers and the inner circular layers. Then you have the submucosal layer where you have that, my enteric nerve plexus. You can also see between the circular and the longitudinal muscle fibers. Then you have in the submucosal layer, then you also have the submucosal nerve plexus. Then you finally have the lumen, and that's where the mucosal layer is surrounding. You can also see the mesentery with the nerve and artery and lymphatics coming to and from. And then on the right-hand side, you have what it would look like under the scopes in a general view. Here, actually, let's go back to this. So this has a much thicker outer longitudinal muscle and an inner circular muscle layer. And I know that, and then I see in the submucosal layer, we have some what look like glands. And our mucosal layer looks thick and it looks uniform, and it doesn't look like there's any like divots or grooves like this. So that is how I would help identify that this is actually probably part of the large intestine, perhaps maybe closer to the smaller intestine, versus the top picture. The mucosal layer looks a lot more like this, and the submucosal layer has some mucus glands, but not a lot. We don't really have a nice thick differentiation between the circular layer and the longitudinal muscle layer, so there's not a lot of movement really happening in this area as much as other layers, and we have a much thicker serosal layer than we do in the bottom layer, so maybe this is a little bit more small intestine, not necessarily large intestine. Now in this picture, in the following slide, on the left, we can see that it's labeled the stomach, but the mucosal layer is a much deeper layer, so not only do we have this going on, but they go even deeper, like this. That'll be really important whenever we start talking about the stomach. Then you can also see a bunch of glands, which is important. Those will be secreting things into the stomach. And then the muscularis layer is really thin, almost negligible. It looks like the serosal layer is cut off there. In the middle, we have the separation of the parts of the small intestine. So starting with the duodenum, then jejunum, then ileum, from top to bottom. And you can tell that they have really, they don't look similar at all, even though this is all part of the small intestine. So being able to identify the differences between each of those 4 layers will be really vital in figuring out where you are in the small intestine. And then on the right picture is just another view of the large intestine. And I can tell that again too because the mucosal layer is relatively uniform. There's not a lot of, or I don't see any, let me zoom in. I don't see any glands here in the submucosal area. The large intestine is mainly concerned about absorbing water, so we don't expect to see a lot of glands here. And the muscularis layer looks really nice and thick, which makes sense because the large intestine is moving quite a bit. Okay. So speaking of movement, let's move on to how we control this whole thing. So, the muscular layers will go through their own spontaneous depolarization. And what that means is, remember our action potentials, so depolarization, repolarization, hyperpolarization, and we reset to waiting for threshold to do it again. So we're going to spontaneously depolarize, which means we are almost consistently depolarizing at the same, or at a consistent rate. And this is a little tricky because you might have one part of the GI tract having the spontaneous depolarizations, but another tract might not have anything going on. So it's kind of a little bit of a gamble of who's doing what when, but the final goal is to, move everything down the tube so it eventually gets coordinated. But since it's spontaneously depolarizing, you don't necessarily know which part is contracting when. That's why we hear like gurgling sounds from our abdomen whenever we're hungry or whenever we're moving. That big Qdoba meal you had last night. Things are kind of going in what we seem to perceive as an unorchestrated mess, but there is some organization to the mess. We call the movements of the GI tract as it's moving down the tube. These are my longitudinal fibers. We call the movement peristalsis. So this is happening mainly on the esophagus, small intestine, large intestine. So these are long portions of the GI tract that don't have any specialization, or I shouldn't say not any specialization, but it's essentially excluding the stomach because the stomach is not straight, right? It's a that's not what the stomach looks like. It's a J-shaped organ, more like this. And so peristalsis is where we're slowly squishing things down the tube, like rolling a toothpaste tube up. This is a coordinated movement. You have to have peristalsis at one end of the tube happen before it continues down the tube. It's not like this part of the tube will happen before this part of the tube. It will start from beginning all the way to end. That's one piece. So that's peristalsis. Now we have to introduce another thing called segmentation. Segmentation is different. Peristalsis is moving things down the tube, but segmentation is focused on constricting portions of the muscle in order to keep things there for a second. So segmentation will constrict off those muscles, those muscles to help keep things within the tube for a period of time. So while peristalsis is focusing on trying to move things down, segmentation will halt the pre-poop, kind of keep it stuck and sequestered in these chunks for a bit of time. This is really important because we need segmentation. This mainly is happening in the large intestine. It happens in other areas too, but it mainly happens in the large intestine. This is really important because then we can have churning and moving of these parts so that we can increase the amount of surface area and increase the amount of contact we have with everything that's in that portion, so that we get the most amount of nutrients and water out that we need. We want to make sure that the whole thing before it leaves is dehydrated as much as possible and is really only consisting of waste. It would be a real inefficient process if we didn't have any mechanisms to make sure that we were able to collect every single nutrient possible. If you think about this is terrible, but if you think about a piece of pre-poop forming, you have easier access to the outside of the piece rather than the inside of the piece. So in order to get to the inside of the piece, you kind of have to break this art up and reshape it so that you get access to that inside piece. And that's what segmentation does for us. So now I have two slides that kind of show you what's going on. This one is more focused on peristalsis. So moving things from left to right, you kind of squeeze one end, but leave the other end open so you can move it down. We also have two waves of peristalsis. One wave will take down the larger foodstuffs or the larger bolus down, And then whatever's left over will go through a second round or a second wave of peristalsis. You can think of this as like eating a piece of cake, and the cake went down, but the crumbs were left behind, but we still want to take the crumbs down with us. We don't want to leave that there to irritate any parts of the esophagus. So it'll go through a second round of peristalsis in order to clear off that extra sludge. Versus segmentation, which is shown on the right, left picture is peristalsis. Segmentation allows us to actually sequester portions of the waste products, so that we can have the greatest amount of turnover, so we can get the most amount of absorption, etc., etc. So how do we control all of this business? Like I said, orchestrate the cluster mess. We have three pieces: local factors, neural mechanisms, and hormonal mechanisms. Local factors, this is more things that are actually contained within the lumen, or within the blood supply surrounding that immediate part of the GI tract. And depending on that composition will affect whether or not we need an increased digestion, increased reabsorption, etc., etc. Some factors include pH, the actual volume. So if we have a lot and then that distends the tube, stretches it out, that can affect motility. It will increase motility. Chemical composition, this mainly has to deal with how the bacteria are interacting with whatever it is that's in the tube. Overstretching. If there's any benefit or irritants, we might release prostaglandins and histamine to promote blood flow and to also promote inflammation, which is blood flow, increasing blood flow. Then we have neural mechanisms. This is where we're thinking about the myenteric plexus and a little bit of the submucosal plexus, but submucosal plexus is more of a local factor piece. The neural mechanisms, those short reflexes, they're named short because they have a less farther distance to travel. So they're like organ, or let me draw a picture: liver, stomach, small intestine. So the short reflexes, it's either within the same organ or nearby organs. Versus long reflexes have to go all the way back up to the brain. So we call that a long reflex. Let's go down to the stomach. And long reflexes have to travel through the central nervous system. Short reflexes stay in the peripheral nervous system. And then we also have hormonal mechanisms. These are only traveling through the blood stream. So remember, all of this is surrounded by a blood supply. So at any point we can have hormones leaking out of there, like they're supposed to, and those hormones can come and actually make contact with the serosal lining of each of the organs, and tell them what to do, depending on what the hormone is. I believe this is your chapter's, not spotlight feature, but it's a pretty large picture at the end of this section, depicting the three mechanisms. I think it does a really good job of explaining what's going on. Hopefully the picture is not too confusing. I like the picture, but maybe it's a little confusing if you're not already familiar with the 4 layers of the GI tract, so become familiar with that first before tackling this. But this concept is really important in order to understand the whole process of digestion from beginning to end. So before you continue on, I would really encourage you to have this down packed. All right, next video will be 24-2.